As we follow then the mechanism of linear DNA transformation, let's go ahead and assume that this is going to result in non-reciprocal general recombination. Now that's a mouthful, but don't worry. What that means, right, is that here when the energy is put in, it actually leads to a change. So we see in this set of circumstances that a recombinant is going to be formed. Now when we look at it being non-reciprocal, of course it's non-reciprocal, right? Because we know that it's only one direction transfer from the donor cell to the recipient cell. So we're looking at an event where the transformation does lead to a new changed recombinant genome for this organism. Now, when this happens, we know that we've got a cell that's in an environment with a lot of free naked DNA. And the first thing that's going to have to happen is that free naked DNA is going to have to associate with the surface of the recipient cell. And that's what it does. And on the surface of that recipient cell, we recognize that the next step will be entry into the cell. So the second step then that we see, the first step of this process is entry. So getting that DNA into the cell. Now, depending upon the organism, this might happen in a little bit different way. But one thing that is universal is that only a single strand of this naked DNA is taken up. So this is a pretty cheesy drawing here where you can see the double-stranded DNA. Of course, it wouldn't be flat out on the page like that, but this gives us an idea of it associating with the surface of the recipient cell. And what's really fascinating is that only a single strand is actually taken up. So one strand is taken up while the other strand is degraded at the surface of the cell. So let's write that down. One strand enters the cell, the other is degraded by exonucleases. And it might be that you're kind of scratching your head right now and you're thinking, wait a minute, how does it enter the cell? I mean, we know that cells are semi-permeable and there is that sort of barrier to entry of just any molecule, and DNA is a large molecule. So this is an area of very active research because we recognize that in certain organisms such as Streptococcus and Neisseria, they have these transporters that span the surface that are actually called competence factors, and they're what allow the cell to bring in that one strand of DNA. In other organisms like Haemophilus, it actually uses a vesicle. It actually forms a vesicle. This is really unusual, and brings in that single strand of DNA. But in either case, the first step here is entry, getting in that one strand of the donor or the exogenous DNA into that recipient cell. So now, once the red DNA, as we're showing it, the exogenous DNA, gets inside of the recipient cell, the first thing that it's actually going to do is it's going to align with an area of homology. And I know you're like, wait a minute, what is she talking about? I can just see Chelsea and Colleen up front and they're just scratching their heads. They're like, what does she mean, homology? That's a fancy, sexy-minded word to say that it aligns in a region that it has similarity to. So if there's a region on the recipient chromosome or the endogenous to which the exogenous resembles, right? It's going to align next to that. And say that in a simpler term, it's going to align by a region of the chromosome that it looks like, that it has a sequence similarity to, or it resembles it. So that's going to be the first thing that's going to happen once this donor DNA gets into the recipient cell. So once inside, the piece of donor DNA is positioned by hydrogen bonding next to a complementary region of the recipient DNA. The two regions are said to be homologous, meaning strikingly similar, right? They have very similar sequences. Once that has happened, this is crazy. What'll happen now is that the donor DNA will literally replace a region of the recipient DNA. So it will not only align next to that region that it looks like, but it will literally replace it. These two steps are called breakage and reunion. So you can kind of guess what's going on there, right? Breakage is going to be the breaking of the recipient DNA. Reunion is going to be replacing that DNA with the donor DNA. So the red, in our picture, the red DNA is actually going to replace a stretch of our purple looking DNA there. So breakage, a nuclease cleaves the recipient cell's homologous strand and releases it into the cytosol where it is literally degraded. And then the donor DNA replaces a region of that recipient chromosome. So we get breakage and reunion. But perhaps this is kind of like you're still thinking, this is a little unclear. Can't we have a picture? So let's take a minute to look at how this happens, sort of a simplified view, a cartoon view of how breakage and reunion do occur. A picture always says 1,000 words. So here we have, once entry has taken place, the red DNA now is free to come and hydrogen bond and align with a region that it looks similar to. From there, that region is actually going to be displaced. So a region of the purple looking DNA is actually going to be replaced by the red DNA. And we're going to see that in this next picture, where the recipient DNA actually is going to separate, and we're going to see it pair with the donor DNA. So notice that the red DNA has literally replaced a region of the purple DNA. From there, the only step left is to get rid of that original DNA. So an endonuclease, right? It's called an endonuclease because it's not chewing from the end, it's chewing from in the middle. So it's going to break these bonds and it's going to release this region of the recipient DNA and it's going to get degraded. So we're going to see an endonuclease nick and displace, it'll be basically lost, and the red DNA will basically replace it. My cat is wanting to be involved in the lecture tonight, so just fair warning if you suddenly see a furry puffball in the view, that is my cat Gabby. She's being a little bit energetic tonight. So, all right. So she wants to know about all of this homologous recombination as well. So that's what this is called, right? Homologous recombination, because it's a homologous strand of DNA, and the regions are similar, right? And then we're resulting in recombination. So resulting in a changed genome after the event. So now we see that, in fact, that changed genome has a region of donor DNA completely replacing the recipient DNA. Of course, now everybody's happy to see that DNA ligase is going to close the gaps. So this is how the process of transformation with linear DNA is going to take place, but we're following it only in the particular fate of the exogenate is actually to be integrated into the host or into the recipient cell chromosome. Marvelous. Let's go ahead and say some words then about the next mechanism, and I know probably Kelsey Adams is pretty excited to hear about this one because we were just talking about it yesterday in office hours, or today, gosh, in office hours. So we're going to look now at transduction, and before we can really get into the heart of transduction, I want to go back to lab for a minute and talk about what are the two different types of phages that we discussed in lab. So for this, I'm going to go ahead and, as you're thinking about this and maybe jotting down the two different bacteriophages that we did talk about, I'm going to bring up my tablet. Perhaps the all people buy moldy lemons mnemonic is going through your mind and you're thinking about, oh yeah, that's right. That was a virulent phage that always went through that lytic pathway. So let's just jot a note of the two types here. The first type that we spent our time on, sorry, my writing is very poor tonight, apparently. The first type that we spent most of our time covering was the virulent bacteriophage. This is the one that I liken to being kind of stupid because it's the one that gets into its host, rapidly multiplies, and quickly lyses that host cell, burning through its hosts like crazy. So that was the virulent phage, and we recognize that it always followed the lytic pathway. The all people buy moldy lemons mnemonic described that pathway. Now, some of you probably recall the smooth criminal, though, that was the temperate bacteriophage. Temperate bacteriophage operate in a very, a a very intelligent way, if you will, because they actually get in and integrate into the host cell chromosome, and then every time the host replicates, they get replicated along with it. So we call that pathway the lysogenic pathway. So that's that kind of intelligent pathway of integrating into the host chromosome. And oftentimes that's a random integration where they just randomly integrate. One of the most famous types of temperate bacteriophage is called the lambda bacteriophage, and it integrates in a random spot within the host cell chromosome. Hangs out there, gets replicated, but then when it starts to get some sort of signal of distress from the host cell, it's like, woo, I'm getting the heck out of Dodge, right? And it excises itself from the chromosome, and then it goes through the lytic pathway. So notice that a temperate bacteriophage, it's capable of hanging out and chilling in the chromosome, but it's also capable of going through that lytic pathway and lysing the host cell when it feels that it needs to go ahead and move along. So what I'd like to discuss then is that the types of transduction relate very much to the type of pathway that's being taken. That is, whether it's lytic or whether it's lysogenic, the type of transduction that we might see is different. So let's go on and look at these types of transduction and then relate them back to what we already know about bacteriophage. So what will often happen then is that after a phage has infected a host cell and it's gone into this lytic phase, and it's going to go through maturation and all of those parts as parts that were made in biosynthesis. They're going to all be packaged, and one of the last things that's going to happen is that the phage capsid is going to be packaged with DNA. But sometimes that phage makes a mistake and it packages host DNA or bacterial DNA in place of its own phage DNA. So we recognize then that this phage now carries bacterial genes in place of where it would have had phage genes. So we often say that that particular bacterial phage is defective because it carries bacterial genes and it really doesn't have any threat. Well, it has minor threat, right, to this cell that it might infect afterwards because it only contains bacterial genes. So phage-containing bacterial genes then go on and infect another cell, and they transfer those genes from the first bacterium to the second. So we recognize that the phage itself is what's allowing the horizontal gene transfer to take place, right? It is the taxicab that's delivering the DNA from the donor to the recipient in that unidirectional manner. So now there are two types of transduction, and depending upon which pathway is being followed, lytic or lysogenic, one or the other is possible. So these two types of transduction are generalized and specialized transduction. Generalized transduction takes place in the lytic pathway. And essentially what's happening there is what I've described, where basically a bacteriophage is trying to put itself all together and it makes a mistake and incorporates bacterial DNA. So with generalized transduction, it's really quite random because when that bacterial DNA is incorporated, it could be any gene, right? It could be any parts, loner DNA out there that gets incorporated into the capsid. So it's very random with respect to that. And I can kind of visualize a lot of you thinking about this right now, and maybe you're thinking, well, how many genes or how much DNA can be incorporated into the capsid of the phage? How much donor DNA is getting transferred from donor to recipient? The answer to that is that it's limited based upon the size of the phage capsid. So, if you've got a tiny phage capsid, it's not going to be able to fit very much bacterial DNA in there. So on average, you're looking at a very small amount of bacterial DNA that's getting in there, like 1 to 2.5% of the total genome would actually get incorporated and transferred to the recipient. So it's a tiny amount that really is getting in there. Now, generalized transduction contrasts with specialized transduction. Specialized transduction can only happen in the lysogenic pathway, because what happens here is that after the phage genome has integrated into the host cell chromosome, then sometimes that phage genome makes an error in excision. So you can picture that it's firmly placed itself into the genome of the host cell, and so it's flanked by bacterial DNA on either side of the viral DNA. So when the virus excises, sometimes it accidentally cleaves at the wrong places. And so instead of excising phage DNA, it maybe excises a little bit of phage DNA, but it excises a bunch of bacterial DNA that flanks the site of integration, and it takes some of the bacterial DNA with it. So this is unique to specialized transduction is unique to temperate bacteriophage and to the lysogenic pathway. So let's read through this. An error is made during the lysogenic life cycle, allowing the phage to take some of the host DNA when it leaves the host chromosome. Thus, in this type of transduction, only a few specific genes next to the point of integration may be transferred. It's not random the way that generalized transduction was. So if there's a specific note that the genes next to a site of integration are taken, you have to recognize that that'll be specialized transduction. We're going to talk a little bit more about generalized transduction and think about that. Before I leave specialized transduction entirely behind, it doesn't hurt to ask the question, and maybe I'm anticipating some of you asking this question, of whether or not the bacteriophage is still functional at all in specialized transduction. That is, because some of it gets excised, right? When it comes out of the site of integration, does it cause damage then to the next host cell? And the answer is generally no. It's generally defective and won't serve any other purpose than just being a transducing particle, that is, being the taxicab that delivers donor DNA to a recipient. Okay, great. So let's look more closely at generalized transduction. Everybody recognizes the all people buy moldy lemons sort of sequence of events that we see within bacteriophage replication in the lytic pathway. And so as we recognize this as being the pathway in which generalized transduction takes place, notice here right after penetration, some of the early genes that are expressed in the biosynthetic phase are those genes that encode for the cleavage of the host DNA. So you notice that the red DNA there is host DNA. So when maturation comes about and the packaging starts taking place, then we can recognize how easily it could be that one of these phage capsids could mistakenly get packaged with the red host DNA in place of the black phage DNA. So that's what we see here, and right here this particular bacteriophage is the defective one. It is the one that is going to go on and possibly cause recombination as opposed to causing viral infection. So we call that the defective bacteriophage. And sometimes, if you want the sexy-minded term, we call it the generalized transducing particle. I'm going to jot that down in case you want to have that in mind, that the other name for a defective bacteriophage is a generalized transducing particle. If I can write in such a way that you guys can even read this, it must be getting too late. Generalized transducing particle. Well, we almost got there. So that's another name for it because it is the particle that is allowing transduction, and the type of transduction is generalized transduction. So it's the taxicab that will deliver the donor DNA, the red donor DNA, to the next recipient. So let's follow this defective bacteriophage on its way to go off and infect another host cell and see what might happen with that DNA, what might be the fate, that is to say, of the exogenate. Sounds kinky. Okay, so of course, again, the stages of adsorption and penetration will occur, but after penetration, the red DNA is going to potentially integrate into the host cell chromosome. It's a little bit different than in transduction, where only a single strand of the DNA comes in. Here in this case, the whole thing, it's a double-stranded molecule. And in fact, when full all-on transduction takes place, the double strand actually fully integrates rather than being a single strand integrating in there. So we can get full transduction where in fact that donor DNA is fully integrated into the exogenous genome, so or the endogenous genome. So the exogenate is integrated fully into the endogenous genome, right? We can also get a process, and often, like sometimes this is called complete transduction, and I'll go ahead and note that on the side here, that this is complete transduction. And then the other possible fate is one in which this is a crazy one. This is where you can see the red donor DNA is just chilling out in the cytoplasm. It's not integrating, but it's not getting degraded. And this is crazy. This is when you get these situations of partially diploid E. coli cells where they're actually potentially expressing multiple copies of the same gene, and that's just nuts. So we see this being something that is sort of sometimes it's a circumstance that doesn't last very long, right? It's not perpetuated, but for a short period of time we get a partial diploid, and this is called abortive transduction. So the cell is actually partially diploid. And of course, everybody knows we can have the energy nothing circumstance where degradation is the fate of that exogenate DNA. So I hope this helps cover transduction, and on Monday, everybody can get stoked because we're going to be talking about bacterial cell sex. On that note, have a fantastic weekend.